Good morning. I'm Lisa Nichols, the Director for Research and Regulatory Reform with the Council on Governmental Relations, or COGR. We're an organization of research universities, independent research institutes, and academic medical centers. And our organization has, over the course of the last year, made an effort to take stock of the resources that institutions offer to raise awareness of and to foster rigor and reproducibility in research, including a survey of our member institutions. As one might imagine, there is a lot of variability in current efforts and practices across institutions, both in terms of how institutions are engaging on this issue as well as the level of engagement. And there's been a lot of discussion on the role of institutions. What is their role in facilitating transparency and reproducibility versus journals and the federal government, and what are the most effective approaches? This symposium and our next panel present an opportunity to hear more about the efforts that institutions are taking or might consider in furthering these efforts. And joining us today for our panel on strategies invoked at research institutions are Dr. Leonard Freedman, the founding director or founding president of the Global Biological Standards Institute, an organization that advocates best standards and practices to enhance the quality of biomedical research. Dr. Brian Nosek, the co-founder and executive director of the Center for Open Science that operates the Open Science Framework, an open source project management repository, and Dr. Ted Taraschi, Senior Associate Vice Provost or Provost for Research Conduct and Compliance at Jefferson, which is implementing the use of electronic research notebooks university-wide. So we're going to get started with Dr. Freedman today. So I'll just give you a brief history. Prior to joining or founding GBSI, Dr. Freedman served as Vice Dean for Research and Professor of Biochemistry and Molecular Biology at Jefferson. He also led discovery research efforts in the pharmaceutical industry as Vice President at Wyeth and an Executive Director of Merck. Before moving to industry, Dr. Freedman was a member and professor of cell biology and genetics at Memorial Sloan Kettering Cancer Center and at the Weill Cornell Medical College. So please join me in welcoming Dr. Freedman.
Thanks, and I'd like to thank the organizers for inviting me here today, seeing some old friends. As was clear from the introduction, I used to be at Jefferson, so it's great to be back in Philadelphia. I have only 10 minutes like the rest of the panelists, and I'm going to focus on one little slice of the reproducibility issue, and that is, I think, issues and hopefully some solution sets around preclinical research, basic research in the biomedical sphere. And Dr. Ioannidis alluded to many of the drivers that are in play that are some of the causes of reproducibility. We published a white paper a number of years ago, my organization, GBSI, and this is a difficult slide, but we're listing based on stakeholder interviews that we held, stakeholders in academia, in industry, in government, foundations, et cetera. And you can see a couple. I'm not going to go through every one of these drivers. There's obviously issues around protocol reporting, reagent variability and lack of validation, which I'm going to focus on in the next few minutes, experimental bias, et cetera. One point that was made a couple of times that I think is important, and I like to always mention because it makes me feel better, is that scientific misconduct is actually very, very rare. So these are issues that I think are solvable. Some are more solvable than others, but the costs to society are extremely high. And one publication I do want to mention that is the only publication I've ever been involved in that had two economists as co-authors, Ian Cockburn and Tim Simcoe at Boston University, was an effort to estimate, at least in one slice of the pie, how much money is being devoted, if you will, or applied to irreproducible research. This was published 3 years ago in PLOS Biology, and we took some fairly simple basic assumptions. One was that in the US in 2014, there's about $120 billion that was spent in biomedical research, that is clinical and preclinical. Half of that, about $56 billion, was applied to preclinical research. Again, that's what I'm focusing on. And if you generously, I think conservatively, take a 50% irreproducibility rate, John had mentioned 70, 80, 90%, which I think is probably closer to reality, then you come up with a number of $28 billion that was spent, at least in 2015, on irreproducible research. Now, that was something that people really focused on when this paper was published, but what a lot of people missed was that we proposed several solutions to closing that irreproducibility spend gap, and they're listed on the side here. And the biggest driver, and you can quibble about whether or not study design is more prominent than invalid biological reagents, but from our perspective, my organization was really focused on standards and how standards can contribute to validation of biological reagents, which we believe was a prominent, if not the most prominent driver of this very, very large number. So if you think about biological reagents, and this was a survey that the scientists published in 2016, the big three, really, at least based on this survey, are in the reagent space, are animal models, antibodies, and cell lines. I'm going to focus again, by way of example, on some low-hanging fruit that can be applied to validation of cell lines. We've also done a lot of work on antibodies, which is a whole nother story maybe we'll have time to discuss in the panel, as well as animal models, equally, if not more important. Cell line misidentification and contamination is very, very common, not surprising to many of you who are in the lab carrying cell lines. It's due to easy cross-contamination from promiscuous growing cell lines such as HeLa, misidentification of lines, not going down the hall and getting a cell line and not validating it. And one estimate is that 15 to 36% of cell cultures in circulation are misidentified. And there's actually a website that a group of cell repository experts have put together called ICLAC. And in that, I went on it the other day, and there are currently 488 documented misidentified cell lines. I think there are many, many, many more, but these are documented. So one could go on that site and make sure that the line you're carrying in your lab is not one of these, because you don't want to publish a paper where it turns out that your study on a lung cancer cell line, for example, may be an esophageal cell line. It's a huge cost to science. And what's interesting is that the majority of researchers, despite our efforts and many other organizations' efforts, including NIH, the majority of researchers still don't authenticate their lines. And we published a survey in 2015 in Biotechniques. We had over 500 respondents, biologists who responded. And the key question that I showed to you is a simple one, which was asked: how frequently do you perform any one of the following species-related authentication tests on your cultured cell lines? And that includes sequencing, STR analysis, which is DNA fingerprinting, karyotyping, which is kind of old school, isoenzymology, and even SNP analysis. And in each case, if you look at the gray, the vast majority, 70%, 74, 75, 80, 85% answered never, 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 never. So that's disappointing and disturbing, but I think an area where there's a great opportunity to close this gap. And I also just wanted to show you that there continue to be papers that are coming out indicating how important this is to the validation and reproducibility of biomedical science. One came out last year around this time in PLOS ONE, and it's got a very clever title, The Ghosts of Gila, How Cell Line Misidentification Contaminates the Cell Literature, get it? And the bottom line there is that they found that over 32,000 articles reporting on research with misidentified cell lines were identified. But what really got my attention is that those in turn were cited in 500,000 other papers. So you can see how this is a sort of a snowball geometric kind of effect. The other came out just last month in Nature from Todd Golub's lab at the Broad Institute, or Dana-Farber, where another issue that I didn't mention is, especially in human cancer cell lines, is genetic drift. And they did an exhaustive study that shows how important the authentication of cell lines that are used in drug screening have to be, because it clearly alters any given cancer cell line's drug response. And again, having been in the pharma world, I am acutely aware of how important that is in early drug screening. So there is a solution, and the reason why I'm talking about a cell line validation is that this is an example of where universities and other institutions can invoke a very simple solution set. And that is that using DNA fingerprinting or STR analysis, which has become a bona fide biological standard, you can actually easily and fairly cheaply authenticate. Now, this is the case for human cell lines specifically, and it is available. There are contract labs that will run it for you at about $100 a pop, and I think that this is a great opportunity for universities and other research institutions to sort of standardize this, either within their institutions, departments, or individual laboratories. And again, I'm happy to talk about that in more detail. My last couple of slides are other areas where I think that there are opportunities. NIH launched, I think as many of you know, anybody who's written an R01 application over the last few years, issued a set of new guidelines in 2015. It's part of a overall rigor and reproducibility initiative. There are many criteria in those guidelines, but I point out the third paragraph here, where, or the second rather, where key biological and chemical resources are not limited, including but not limiting to cell lines and chemicals and antibodies, need to have an authentication or validation plan in their grant proposals. And I have to confess, I was a little skeptical. I thought this was a little too vague in general when the initiative came out. They're not scored as part of a study section. They're actually considered once the scoring takes place within a study section process. But I've come around, and because I think it's raised the awareness level. Any investigator, and many of you in the audience who've had to sit down and write an R01 and have to actually come up with an authentication plan, at least are thinking about this. So minimally, it's forcing people to take notice. And then the final thing, and again, I hope we can talk about this, and it has been alluded to in passing this morning, is the importance of training. Training our students, our postdocs, our junior faculty on all of these issues, not just authentication, validation, but study design, statistics, everything you've heard about today. And I think that is, again, another very, very important responsibility and opportunity for our academic institutions. And I can tell you again, and I'm sounding like a shill for NIH, Dr. Tabak, please note, but there are a number of RFAs that have been issued by NIH in the R25 mechanism. My organization, GBSI, has received 2 R25 training awards with a focus on reproducibility, one generating video training modules on cell authentication, the other on aspects of study design. And I can also tell you in closing, I'm actually literally on a study section tomorrow on a second R25 RFA that's been focused on this this training module initiative on data reproducibility, and the pay lines are a hell of a lot better than the pay lines that you're used to for R01. So again, I think a tremendous opportunity for the Philadelphia and nationally universities to initiate training within your institutions that have a focus on how to do science, not in the sort of apprenticeship mode that we've all grown up with, but in a sort of more didactic and focused way. So with that, I think I'm going to stop, and thank you.
All right, thank you, Dr. Freedman. Our next speaker is Brian Nosek. Dr. Nosek, prior to founding and becoming the executive director for the Center for Open Science, or in addition to that, he's also a professor with the Department of Psychology at the University of Virginia. He received his PhD from Yale University. Dr. Nosek, in his role at the University of Virginia, investigates the gap between values and practices, such as when behavior is influenced by factors other than one's intentions and goals. He was named one of Nature's 10 and to the Chronicle for Higher Education Influence List. Please welcome Dr. Nosek.
Last week, my fourth grade daughter, Joni, brought home her assignment of learning what science is all about. And it described a lot of things that are familiar. Scientists are curious about problems, they make observations in the world, they generate a question that they want to investigate, they design a way to study that question, they do collect some data, then they write about what they found and they share it with others. And then other scientists talk about it and da-da-da-da. Nowhere in her assignment did it say scientists spend all their time writing grants, they have to churn out as many papers as they can, and so it took me a while to convince her that science wasn't like that at all, and describe all the things that science was, and she lost interest about 5 seconds into that conversation. See, she's still upset about it. The challenge, of course, that the ideals of how we think about science, how we educate what science is as a methodology, are sometimes in conflict with the realities of what it takes to have a career in science. And that potential gap between what we want science to be and how we have to do it in order to succeed involves a lot of stakeholders that drive incentives. What does it take for me as a researcher to survive and thrive in science? And the major stakeholders in that are the granting agencies, right? Who gets the money to continue doing the science? It's the journals and publishers who gets through the pipeline in order to present their work in an outlet that will advance their career in some way by being prestigious or getting attention or otherwise. And then the institutional responsibility, as we were talking about for this panel, is who gets hired and who gets promoted and tenured. And what are the things that are expected of scientists in order to get hired and promoted and tenured? And that's what I want to focus on as the problem given the panel discussion this morning. And as we are aware, and as John's opening keynote put a point on, is that publication is really the currency of scientific advancement. Right? I need to publish as much as I can and get in prestigious outlets in order to advance my career. And so what is it that institutions do to decide how it is that they shape what researchers do in response in order to get the rewards that they need for the institution, getting that job and keeping that job? And there are a couple of core problems in how it is we operate hiring, promotion, and tenure that are addressable, but if they're not addressed, continue to perpetuate these challenges of reproducibility because the incentive structures of getting the outputs sometimes lead to behaviors that reduce the credibility of those outputs. So problem number one for promotion and tenure is that the policies for promotion and tenure are often vague or opaque. Right? Researchers themselves don't know precisely what they need to do in order to achieve promotion and tenure. And there are very good reasons for that to be the case. Right? We don't want to constrain scientists to do their scholarship in a particular way. publish X number of times, or publish in X outlets, or do these kinds of methods, right? Part of scholarship is to try to keep it as open-ended as possible. And so we use terms, if we communicate anything at all about what does it take to get tenured, we use terms that are very general. You have to have high impact. You have to change understanding. You have to promote new knowledge. And so in some ways that's appropriate. But because it's vague, because I have to make decisions of what are the behaviors I'm going to do to try to get advanced, and saying I need to have high impact, well, I don't know quite how to do that. But I do know how to write papers. I do know how to write grants, right? So that I can fill in the gaps of ambiguity with the concrete behaviors that I perceive as the ways that I get advanced in my career. And so to the extent that we are vague in how we describe these goals, we will provide an open interpretation, conspiracy thinking about what it really means to actually get advanced in the career. And simultaneously, there are things that we do in the promotion and tenure process that implicitly communicate expectations that may not be aligned with the values that we actually have in deciding who to hire and who to promote. One example, I went up for full professor a few summers ago, and the administrator said, please print out all of your papers and give them to me so the committee can review them. Now, going up for full professor, I at that point in time had 100 papers. So I said, you want me to print out 100 papers and give them to the committee so that they can review them? She said, yes. I said, what are they going to do, weigh them? Right? It is a nonsense request, but it does communicate something about what the institution values, right? What it communicates implicitly is that volume is what matters. And obviously, no one on my department tenure committee, nor on the dean's level, nor at the provost level would say, all we care about is quantity. That's what we want. None of them would say that. But because they asked me for all of them, because I knew in advance I had to print them all out, that's the message that I receive, that in fact, that's what matters. And so there are lots of things that we can change in how it is we present what one needs to do. Right? So if the admin had instead said, when I walked in the door of the institution on the first day, when you go up for promotion tenure, you're going to print out 3 papers, and you're going to give those to the committee, and they're going to actually read them. And that's going to be the basis of evaluation. I would have had a very different mindset about what it means to try to succeed in this context. And so the details actually really matter because because it's open-ended, we have to fill in the gaps of, given my area of scholarship, what do I need to do to have impact, change people's understanding, become internationally acclaimed, whatever it is that are the vague terms. The subtleties are things that are going to make a lot of difference. Last point on the problems is that of all of these different stakeholders, funders, the journals, and the institutions, the hardest incentives to change are at the institutional level. And the reason that they're the hardest to change is because administrators have very little control of how it actually happens. Sorry. The reason that there's very little control is because the process of hiring, the process of promotion and tenure, is reconstituted every single time it's done. There is a new committee for every hire that happens in most departments. There is a new committee for every promotion and tenure case. And so it's very hard to establish strong institutional norms and standards and have those apply consistently over time, because this unique constellation of an individual committee may have different ways in which they approach the problem of who is it that we're going to hire and who is it that we're going to tenure. And so disseminating what the standards are is an ongoing challenge because it has to be done every time, and it requires getting all of the senior faculty or decision makers on board with the process that we are going to implement. Whereas at a journal, if the journal creates a policy, then all authors follow that policy. If a funder creates a policy, all grantees will follow that policy. So a couple of things that, despite those challenges, are solvable in how it is we approach this institutionally. First is improving explicitness, right? There is no benefit to saying nothing for how it is one succeeds, because the actor, the individual researcher, has to fill in the gaps. So the more that we can provide of explicitness of what it means to be successful, what it is we evaluate, what it is we don't want. We don't want you just to produce and produce and produce. We want you to think about quality. The better off researchers will be in figuring out how to translate success criteria to what they do on the ground. The second is to actually incorporate evidence of rigor and of transparency and reproducibility into those policies and provide that training context for how is it that the evaluators that are dealing with that decision process can handle that information. How is it they can incorporate that into decision making? And then the last is actually investigating the problem and sharing that knowledge. There is very little cross-institution communication about what the policies are for tenure and promotion or hiring, what the outcomes are, and how it actually gets implemented in practice. And just sharing that information would have a couple of benefits. One, it can start to identify best practices that may occur across institutions. And two, it can actually provide a database to see if the theories that researchers have are actually the realities. Is it the case that the number of publications is a strong predictor of actually getting tenure? We don't actually know that. There isn't enough data to investigate that question because we don't have an ethic of actually investigating this as a problem. So if we actually study it and see that in fact it predicts 50% of the variance of who gets tenure by just how many publications they have, then both the institutions could say, oh, that's not how we want to make decisions. And researchers can say, oh, that's how they make decisions, and be informed by that. So having the knowledge is the best way to intervene so that we can align the values that we have for science with the daily practice of what's rewarded for scientists. Thank you.
Thank you, Dr. Nosek. Our next speaker is Dr. Ted Taraschi. Again, Dr. Taraschi is a Senior Associate Provost for Research Conduct and Compliance at Jefferson. Dr. Taraschi oversees the Office of Research Conduct and Compliance and facilitates the conduct of ethical and regulation-compliant research at the university. So, Dr. Taraschi.
Thank you for the introduction. I'm actually well placed because I had an NIH-funded lab for 35 years. So as I've transitioned into administration, I sort of know both worlds. So I think I'm in a good place for that. So at Jefferson, we've developed comprehensive policies and programs to respond to recent mandates from federal sponsoring, research sponsors. And I'm going to discuss two of them today. And the first is the responsible conduct of research. And what I could say is that, and I was just talking with my colleagues here, the way it goes is you go to a postdoc somewhere and you're at a good lab with a good reputation and everybody knows the place and the people, and it's assumed that you know how to do everything. You know all the aspects of being a successful scientist, and in fact, I think that's a rarity rather than that's the norm. So we've developed a responsible conduct of research program. I think ours is different in that our provost is requiring that all the faculty have to do this as well. At most places it's restricted to students and postdocs. Here all the faculty are going to have to do it every 4 years. Just to remind everybody what those components are, they're shown here: authorship, collaborative research, conflicts of interest, data management, mentoring, peer review, research involving human subjects, research misconduct, and using animal subjects in research. So we've established new policies and guidelines for the training of our whole research community, as I've said, and so everybody's going to have to take 9 modules of the city program every 4 years. And I'm fortunate that I'm an institution where the provost is really supportive of this, and it makes it somewhat easier. The second area is to develop a campus-wide scientific rigor and reproducibility guidelines. So as we just heard from Dr. Freedman, and I think he told me that PLOS paper that he had 80,000 views, which is incredible. It's been estimated that I think he said $28 billion in preclinical research has been deemed to be non-reproducible. That's a complex issue. It's due to a number of factors, including insufficient detail or documentation in research data records. So that, as we've heard from Dawn, as she sort of charged the group and others here, the impact of irreproducibility in research is, spans from potential degradation of public trust to economic impact. So one of the ways Jefferson has decided to deal with this is to require that all in the research community will be using an electronic research notebook. So what are the obstacles to doing this at an institution? The biggest one is, if you do clinical trials or if you do research in the pharmaceutical industry, like having an electronic notebook, having your data for the day signed off on, there's templates for how to do things, it's highly audited and monitored. That's not the case in basic research, right? And in basic research, everybody thinks their way is the only way. Nobody can share anything. You can't share. There's the concept of having a template for things like how to do a Western blot or how to choose the number of animals to do research. Everybody claims nobody can agree on that, and I don't think that's the case actually. So what are the biggest obstacles from an institutional perspective to do this, to implement an electronic research notebook? You're asking the PIs and the research community to change their behavior. People have, used paper notebooks, and what we found when we looked at data uses and storage practices, most people have stuff in 15 different devices in 15 different places, not annotated, not discoverable. And I'm also the research integrity officer at my institution, and I can tell you that I've been very busy with all the outside groups that are looking at your publications, and it's really important to have the original data. So what we're also finding too is that the state of the IT infrastructure in laboratories is not always great. Some people were using wireless that wasn't meant to transfer big data. People are not connected to the internet because they have old computers that are out of date or whatever. So there's a lot of maybe expensive. An added bonus of having an electronic research notebook is that you're going to upgrade the infrastructure in your laboratory. So there's benefits both from the institutional perspective and from the user perspective. From the user perspective, you can be organized and you can find things. And when people leave, you can find it. It's documented. It's discoverable. You don't, how you never find the people that left and their data, and you're going to write that paper and they left 2 years ago, and you're like, well, who did that? And you spend more time trying to find all that than you probably did doing the research. So you need to develop and sustain partnerships for the implementation of continuing education and customer support. At Jefferson, it's my office, it's the office of the provost, it's library services. We have Gary Kaplan sitting here from our library, and it's IS&T. So it takes a combined effort to do this. One of the big challenges that we have, and I don't know how we're going to deal with yet, is monitoring and auditing. Because everything, your notebook will only be seen by you, but we got to make sure that you're actually not using it just as a great big repository to just throw everything in there unannotated on this, it has to be sort of set up and used in the right way. And I think that's going to require consistent training and education, which we're in the process of. I think we expect, and I've spoken to others at other institutions, that this will be an organic process, and it has been at Jefferson so far. But I think in the end, there may have to be an institutional mandate to make this work. I just saw that Partners Health, I think, is the first one in the US that will use the mandatory use of an electronic notebook by October 2019. I think Jefferson will be the second. It's mandated in Australia and other foreign countries, but in the US it's been slower to be mandated. So here's our roadmap. We're in this about a year and a half right now. So again, being the research integrity officer and speaking to the provost back in, the beginning of 2017, I said, you know what? I've been meeting with people trying to find original data, whatever, and I'm not really that happy with the status of that in a lot of instances. That's not implying anybody did anything wrong. It's just they can't put their hands on things that they did 3 or 4 years ago, always that readily. So I got designated as the executive sponsor in March of 2017. The first thing I did was assembled a research data management task force to look at what was this current state at Jefferson. And that group, we looked, I think there were about 18 companies that were in this electronic research notebook business. I think we had 3 of them on campus, and we decided we chose Lab Archives as the ERN at Jefferson. We began a campus education program in the end of 17. In the beginning of 18, we formed an executive committee. This really needs somebody to own this. I say it, and I don't mean to offend anybody in the clinical world, but this is kind of the Epic records for research. You know, in the medical community, they have Epic. This is sort of our Epic. And it takes a lot of coordination and a lot of resources and dedication to get this right. We formed a project team we meet constantly. I, as the executive sponsor, have gone out and met with all the departments and the department chairs and the people in the departments and gone to all those leadership type meetings, you know, executive councils, provost council, just to keep up with it's not going to go away. We're going to keep moving forward. It's a sustained effort. And we're going to keep at it. We got 60 beta testers engaged in March of 18, testing Lab Archives. What are their experiences? What were they finding the best practices? What were they finding the deficiencies to help us move forward with the project? We just had the official campus launch in the end of September. Lab Archives was on campus for 2 days. We ran basic and advanced sessions. This also has an education module. We teach like laboratory sciences, biology, physics, chemistry, etc. And that was highly successful. We're now in the process of actually going to individual laboratories to assess and update their IT infrastructure and computers. Because we're finding, you know, people have computers that are old, that are not in the network, that have operating systems that work for them but can't be updated. And so if the idea is you want your research to go right from instruments right into Lab Archives directly, which it can do very easily, those computers have to be in the network. So that's a bit of a heavy lift. And as I said, our provost expects that all in the research community were using this Lab Archives by October of 2019. So how are we doing? So like I said, we had 60 beta testers, and that was from a group. We could have had way more. I actually wanted to pick the doubters first. I wanted to pick the hardest people to see if we could convince them that, you know, that would be great for growing the process. Like I said, the company Lab Archives has been, I'm not endorsing anybody. I think Penn is also using Lab Archives. They've been incredibly supportive, working with us, and they're growing just like we're growing at the same time. Our training and support is currently being provided by our library services and sort of in a partnership with my office and other offices now, but eventually it will move to the library. Gary Kaplan's here if anyone wanted to talk to him about that later on. And we currently have over 200 notebooks that been established, and we can actually, Jessica Gutierrez is back there, she's the program manager, she can actually look and see the activity that's going on, and these are notebooks that have been established that people are actively using and transitioning away from their practices into using the electronic notebook. And with that, thank you for your attention.
We have about 20 minutes for questions if you want to start coming up to the microphones. I have one question. So you mentioned, Dr. Taraschi, that you approached the skeptics first. What type of resistance are you finding from faculty? Particularly, is there resistance in terms of using one product, for example? And what are you hearing, and how are you overcoming that resistance?
Is that on? Yeah. So the resistance is people don't want to change, really. I mean, that's really the real, why I do it this way, and I'm like, well, okay, well, let me see how you do it now. Like, let me see what your records look like. And it's often a mixed bag, to be honest. And so I think it's just that. People don't want, there are some people that have used alternatives, like I think they're called Evernote or some other, but the one that we picked, that I think it timestamps the data, nothing's deletable. So like from an intellectual property point of view, it like documents what you've done in real time. And I think it's a great tool for running your lab, for sharing and collaborating, and which is where science has become. And also, you know, people come and go, but you can hashtag something, and years from now you can find it, and find it like that, as opposed to thumbing, like when Len and I did science, you know, we'd have 5,000 binders with Western blot films and all sorts of stuff that would be impossible to find. So I think once people see the power of the system, when you actually spend time with them and you show it to them and you give them working sessions and demystify it a little bit, that resistance goes away.
Thank you. Question over here?
Yes, hi, I'm Mitch Mortonford with CHOP. Before that, I was on the Jefferson campus with neurological surgery and then the Rothman Institute. And I was thinking about my experience working with medical students and residents whose goal is patient care and who are involved with research as part of career advancement. What strategies would be in place for working with them to improve research quality and output?
I'm not, it's an interesting question. I don't think it would be fundamentally any different in terms of the approaches to training our trainees, whether they're medical students, postdocs, graduate students, in the fundamentals of doing good science. Obviously, their career goals are ultimately going to be different, but you know, when I was at Sloan Kettering, I was on the MD PhD admissions committee and steering committee, and you know, had a lot of opportunities to speak to aspiring MD PhDs, and one of the things I always asked them was, you know, eventually most people have to make a decision. You're either gonna, you know, focus on patient care, or you're gonna go in the lab and do research. Very, very, very few people get the opportunity to do both. But I think that I don't see necessarily making a distinction in the training years on teaching good fundamental aspects of research, whether you're going to ultimately focus on patient care or ultimately going to be in the lab doing research. Anybody?
Yeah, I agree with that, in particular that the conduct of research has the same issues on methodology as the consumption of research. And the training on methodology is in great need of enhancement, because there are a lot of signals in papers about the degree of uncertainty and the likelihood that the effects are not really reliable. But if you're not trained in identifying those things, it's very hard to make assessments either for what to do as the next step as a research agenda, or how to translate that into clinical care. And so the same issues of understanding the methodology, I think, arise for both.
If I could say too, at Jefferson, they revised the medical curriculum, and every medical student has to have some kind of a research experience as part of their medical school experience.
Thank you very much.
All right, over here.
Yes, Marcia McNutt from the National Academy of Sciences. Because these topics have come up, particularly in this panel, I wanted to mention two upcoming activities at the National Academies that I think might be of wide interest to the people at this meeting. They're both workshops, and therefore I'm hoping that there'll be a lot of interest from everyone here in attending them. The first is looking at the tenure and promotion policies at universities, and with particularly with respect to whether they currently are constituted in such a way that they are appropriately encouraging some of the policies and practices that we recently have thought would be good to have, things like mentorship of young people, open science practices, and sharing of data, reproducibility, et cetera, or are some of our tenure and promotion policies out of date with respect to what we think will make the best possible research in the future. The second workshop is a little broader, and it's looking at the overall culture within the research enterprise and whether that culture is inadvertently promoting some of the counter norms of science rather than encouraging the norms of science that we actually believe will lead to the strongest and most trustworthy research enterprise that we want to have. So I urge everyone to be on the lookout for the announcement when we are requesting people who would like to attend those workshops, because I think they will be of interest.
Can I make a comment?
Sure,
So
absolutely.
I'm very interested in the whole issue around incentives as it relates to reproducibility, as it relates to academia, and I think Brian gave a beautiful summary and of the challenges and the issues, and Marcia, it's really great that the National Academy is going to address it, because I think what's been missing up until now, with some very important exceptions, and I think in particular Glenn Begley, who's a friend of many of us here, has talked very loudly about so-called perverse incentives that are in play. And I'm going to be provocative here, because I was a professor and tenured for many years, and I was a vice dean for research for several years. I'm not convinced that people here in this audience are necessarily convinced that those incentives, those cultural changes that you're alluding to, and Marcia and Brian, I'm not completely convinced that that's going to change anytime soon. And part of it is, again, a good part of it, I believe, is related to the incentive structure, or as Glenn calls it, the perverse incentive structure. So I'm going to be quiet now, but I'd love to hear more discussion about that today, because you guys are in a very unique position to address that.
If I can make a comment too, back to your earlier question, actually. When I interface with the faculty, I said, you're teaching these people. Like, if you have bad habits, they're going to have bad habits. If you have good habits, they're going to have good habits. So, it's really incumbent on you to take an active role here and not sit back and you're training the next generation. You have a responsibility. Like, it's part of doing research is training the next generation. And I think, it's people stopped and thought about it. And like, we're so busy trying to get those publications and trying to get that impact factor up and trying to get those grants. You're training the next group of people. So you have a responsibility, as I see it.
Okay, thank you. Next question, Dr. Tabak.
Yeah, Larry Tabak from NIH. So I'm going to dovetail on the many comments about promotion and tenure decisions. Prefacing it by saying that the plural of anecdote is not data, but I do want to reflect on your comment, Brian, about should I print out all 100 papers? I still run a little lab, and occasionally I'm asked to comment on tenure and promotion of colleagues in my research field. And the provost of the university asked if I would do this, and I said, great. And they sent me, I don't know if everybody in the audience knows what a baseball card is, the equivalent of a baseball card. And on it was the person's name and their H factor and their G factor and their Q factor and their MOUSE factor, okay? And all the information was just on the card, okay? So I wrote back and I said, That's great. Where are the publications? Because, I mean, I could look them up, but I was sort of hoping they would send me PDF. And the response I got from a provost of a major university was, why do you need those? Wow. Wow.
Yeah, there isn't much to say in response to that. Oh, that's amazing. But it is I write a lot of tenure letters now, given where I am, and what is, I guess, encouraging is that there is a wide range. So there are some institutions, some places that seem to just be about metrics and not about substance, and there are others that say, here are the 3 papers. We're not even going to send you the CV. What we want is your comments on these 3 papers. And that diversity of how tenure letters get recruited is invisible to everybody except the tenure writer. So I think that's the thing. If we can make that explicit and that institution can see that no one else does it like that, or at least no one else is willing to admit that they do it like that, would be a very strong normative mechanism for normative change.
Question over here.
Ellen Walker from Temple University. I'm a chair of the Department of Pharmaceutical Sciences, and thank you for a very interesting panel. But one of the things I haven't heard too much of yet is something that I run across a lot, and I'm in the School of Pharmacy. I'm an academic scientist, have been for many years. I dovetail with a lot of colleagues who have come from industry, and now they're academic scientists. And they have to come up for promotion and tenure. They have to train their students. And things that keep coming up is they have a lot of intellectual property. And so I will say, well, you are great. Yeah, you have 3 SBIRs now. You have 0 publications. I can't publish because of IT. And so I'm noticing more and more frequently that, why don't we share all our data? That's kind of where I grew up. But I also realize there's now another set of incentives, i.e. a company within a university, IT within a university, and the university has just as much stakeholding in holding back that publication as the investigator does. So how is an institution and how is a chair supposed to respond to those kinds of things when I will have to put up someone who will have been at a university with grant money, but maybe not publications? So their weight will be very low that way. They count the money, it'll weigh heavily. They count the publications, it'll be light. So that's something I wanted to put to the panel. Thank you.
I can just say, at my institution, that's now become a consideration for promotion and tenure, the innovation component. And Dr. Klasko is about as innovative and disruptor there is. So it's become part of the equation. And again, from my experience, and it's not the same everywhere, but you're kind of like a pie, and teaching is a part of the pie, and service is a part of the pie, and publications are part of the pie, and I think you'll get some leeway in those areas if you're a good citizen in those other areas. That's my experience at my institution.
A general comment on open science in relation to this is that it's often communicated as if everything must be open, and that's the only right way to be. And I think that's the wrong message, right? The right message is open should be the default, meaning we assume we're doing work for the public good and that the things that we produce are public goods. That's what science is about. But there are lots of reasons to not do it. And those reasons include ethical issues of sharing, privacy and otherwise, and intellectual property, because there are very many legitimate uses to promote acceleration of solutions that involve commercialization. And universities are taking that on as part of serving the mission of an institution. It's not just public dissemination of information, but also the seeding, the catalyzing of new solutions in a commercial sphere. And I think that's totally okay. Issue is explicitness, is being transparent about what is the purpose of this line of work. And that may be that some labs are all about commercialization and that's what they do, and maybe some labs are all about public good scholarship, that's what they do, and some labs it's a mix. As long as we know which it is, and that's something that's stated of, we can't share this because X, and this is because Y, then in terms of the open science hat, that's fine. They're stating what their values are. The reward system then is that extra layer, is the institution prepared to say, we are doing this, we are encouraging it, are we providing the incentives in the right way to reinforce it, balanced with the values that we have as an institution? Because we could just transform the institution, say we're all about commercialization. What we are here is an R&D facility to make as much money for the university as possible. Probably institutions won't say that as what their value is, but they might be willing to say a portion of what we do is for that in order to support the public good programs that we pursue.
Okay, this will be our last question from the audience.
I'm John Epstein at the Perelman School of Medicine, where I'm the Chief Scientific Officer. Some of the discussion worries me a little bit, and that is that we need to, as you're pointing out, remember that we're universities and academic institutions. And while I think it's fine that we value innovation and commercialization, we can't do it at the expense of disseminating knowledge. If people want to just protect their IP and make money, they should go into the private sector to do that. And I think the universities have a role in facilitating the open communication exchange of ideas. We want our investigators to be innovative, but I think we all need to band together and make some common rules about where the red lines are, because it's hard to do it as one-offs, one institution at a time. It's easier if we all agree that there are certain deals we won't make, like agreeing not to publish our results. So it's not a question, but food for thought, perhaps.
I just have one last parting thought. You talked about changing institutions' policies with respect to hiring, tenure, promotion. How would you factor in peer review, just in thinking about, how we value publications over, reproducibility, data sharing, and mentoring? Just wanted to get your thoughts very quickly. We have about 2 minutes.
So from my perspective, peer review is essential because that is the mechanism of how science makes progress, right? We aren't individual actors of the knowledge bastion. You just have to ask me what's true and what's not, and that's what advances it. Rather, we advance through consistent critique of each other's work. The relationship with openness is that in order to be able to evaluate the quality of someone's work, we have to actually be able to access their work. John used the phrase, the paper is the advertising for the research, not the research. I think that's a very important part of understanding how peer review is effective and ineffective. We can't address the challenges of research quality effectively if we can't see how the research was done. And so I think peer review plays an important role in seeding both credibility indicators, did experts evaluate this well, and in identifying the limits of what peer review right now can accomplish given what's not available for peer reviewers to evaluate.
And I guess also with respect to a focus on publication versus, other, work that fosters reproducibility and transparency.
That's right. You can reward more things if you're rewarding transparency and reproducibility other than just the publication. It's a great point.
Okay. Thank you. All right. Well, thank you. Please join me in thanking our panel.